So without further ado, I'll pass over now to Moe Moe Kyi Win. She is a final year PhD student at Kasetsart University in Thailand, and she's going to be presenting her work on QTL-seq, finding a key gene for bacterial leaf streak resistance in rice. So Moe Moe, over to you.
Thank you for the introduction. Let me share my screen.
That's perfect.
All right. Okay. Good evening here in Thailand and good morning in UK. Today I would like to talk part of my PhD studies, which is to identify the key genes responsible for bacterial leaf streak resistance in rice by using QTL-seq approach. So in rice, there are two major bacterial diseases. The first one is bacterial leaf blight, BLB, which is caused by Xanthomonas oryzae padova oryzae, or XOO. And the second one is the bacterial leaf streak, BLS, which is caused by Xanthomonas oryzae padova oryzicola, XOC. When the rice plants are infected with BLB, we can see the phenotypic symptoms like yellow-orange stripes on the leaf blades or on the tips, but with a wavy margin. But for the BLS symptom, we can normally see as a small water soluble linear lesions, which can sometimes be translucent. And these streaks can be 1 to 10 centimeters long. Although they share the same species, the way they infect the rice leaf is different. In the case of XOO, they enter to the rice leaves through the hydrodotes and they try to invade the vascular bundle. But for XOC, they enter through via the stomata or other natural openings, and they try to multiply in the stomata cavity and trying to invade the spongy mesophyll cells. So the BLS is mostly limited in tropical and subtropical Asia. It is also regarded as a potential bioterrorism agent in the States. Compared to BLB, the yield loss due to BLS infection is not that serious, but under the very favorable condition, the yield loss can be up to 32%. And BLS resistance in rice is a quantitatively inherited trait. However, compared to BLB, there are just a few number of R genes. Resistant genes have been identified so far. There are two dominant resistant genes, XO1 and XO2, which are encoded by nucleotide binding leucine-rich repeat proteins, and three recessive resistant genes, BLS1, encoded by MAPK6, and BLS2 and XE5. And yes, of course, there are a few number of defense-related genes containing different kinds of transcription factors and antioxidants-related genes and plant defense-related genes. Overexpression of or suppression of these genes give resistance to BLS. So from all these genes, XE5 is now standing as the one of the major genes using currently in the current breeding programs. So I would like to give a few information about XE5. So XE5 was founded as a QTL on chromosome 5, but later narrowing of the QTL region by using chromosome substitution lines gives that there are two nucleotide changes on the resistant lines, and that give resistance to the XOC infection. And that gene encodes gamma chain or transcription initiation factor 2A, which is exactly the same gene that gives resistance to BLB as well. In our lab, the Rice Science Center, Kasetsart University, we also found XE5 as a major gene controlling for most of the XOC isolates in Thailand by using our rice germplasm with the GWAS approach. From this study, the authors also found out that there are some genotypes which are listed here. They show strong resistance to most of the XOC isolates, but they don't have XE5 confer allele or any other known R genes confer allele. So we think that there might be an alternative mechanism to XE5, and using of one of these genotypes as a resource or planting material might be an alternative way to find the new genes. So in this experiment, we use QTL-seq. QTL-seq is an approach to identify the QTLs by using whole genome resequencing of 2 DNA bugs, which involve the contrasting phenotypes of our trait of interest by the next generation sequencing technology. So to start the QTL-seq, we have to get the mapping populations like F2 populations or BC1F1 populations. This is the example from Takagi et al., in which they have a mapping. They developed a mapping population, and from this population, they selected the plants with the shortest plant height as a one bug, and the plants with highest or tallest plants are selected as highest bug. And these individuals are aligned to the reference genome to get the SNP index, and the other SNP indexes to get the QTL and candidate genes controlling our trait of interest. So in my case, I use F2 population as my segregating population. So to get F2, I have HCS variety, which is susceptible to XOC, and NDCMP49, which was recommended from our previous GWAS study. We use it as a resistant parent, and we use F2 for phenotyping. This is just a general workflow of how we phenotype the entire F2 population. We use the bacterial. We spray the bacterial suspension to the 3 to 4 weeks old F2 seedlings, and 2 weeks after inoculation, we recorded the disease score of each individual plant by using a standard evaluation system developed by Erie. And then we selected the plants which are mostly susceptible bug, and the plants which are most highly resistant as resistant bug, and these individuals are sent for sequencing. And then after we got the whole genome resequencing data, we use this pipeline, like aligning the sequence data to our reference genome and SNP calling, and calculating a SNP index and the other SNP indexes, like this plot, and we got QTL. And so from the QTL-seq, we got an interesting candidate gene, and we would like to see the expression fold change of these candidates by the transcriptome analysis by RNA sequencing. In this RNA experiment, we use the same genotypes, NDCMP49 and HCS, and we use the same XOC isolate, but for the inoculation, we used injection by using needleless syringe, and follow this pipeline for transcriptome analysis. So after crossing, we got F1 plants. We also did the marker assisted selection in F1. Here, as you can see, the phenotype of F1 plants are kind of similar to susceptible parents, so which means the the genetics of resistance in this population might be governed by the recessive loci or recessive gene. And then here are the phenotypes of the individuals that we selected for susceptible bug. Also, the phenotypes of the plants that we selected for resistant bug, along with the phenotypes of the parents. And then this is the distribution of all my F500 F2 plants that I did for phenotyping. So here, score 1 represent resistance, and score 3 represent moderately resistant, score 5 represent moderately susceptible, score 7 means susceptible, and score 9 gives highly susceptible. So we selected 30 plants with score 1 as resistant bug, and 30 plants with score 9 as a susceptible bug, and these are sequenced and aligned to the process with the QTL-seq pipeline. And then we got a very we got a QTL with a sharp peak on chromosome 2. So the QTL the region is quite huge. So then we try to locate the SNP with the highest delta SNP index value, and we try to span 200 kb up and down of this SNP with the highest delta SNP index. And finally, we got 22 candidate genes in this 200 kb regions. And then we try to see the expression fold change of these 22 candidate genes. I forgot to mention that in the RNA-seq analysis, we collected the leaf samples at 2 time points, 0 HPI, 0 hour post inoculation, and 9 HPI, 9 hours post inoculation. So in this heat map, the heat map shows the expression fold change of within the genotype and also between the genotypes. Here in the first column, we have the expression of N genotype, which is our resistant parent at 9 HPI and 0 HPI. And the second column is our susceptible parent H CS at 9 HPI versus 0 HPI. And the third column shows the comparison between resistant and susceptible parents at 9 HPI, and the last column is the comparison of 2 parents at 0 HPI. So from all these 22 candidate genes, we have OS02 G063366 shows very high upregulations in the resistant parent at both time points. So what is that gene? That gene encodes protein kinase or catalytic domain containing protein, and according to the annotation database, it says this gene encodes wall-associated kinase 16. As we can see here, at 0 HPI, the expression of this gene is around 9-fold upregulation in resistant parent, but after 9 hours inoculation, the fold change was increased double, around 15 times upregulation in the resistant parent. To confirm this candidate gene as our best candidate, we also developed marker in one of the SNPs that we got from QTL-seq, and our marker development and validation in F2 individuals also shows very high phenotype variance explained, which means this marker is also reliable to use for our future breeding program as well. And we also want to see the possible use of this WEX16 in the wider germplasm. So we did haplotype analysis by using 211 rice accessions. Actually, we were able to identify 16 SNPs in WEX16, but here for the haplotype analysis we use I think 12 SNPs, because the rest of the SNPs they have the synonymous nucleotide change, so we didn't count these SNPs. So from this analysis, we got 6 haplotypes or haplogroups. Only haplotype 1 shows a disease score range of 1 to 3.8, which means the phenotype is like resistance to moderately resistance. And this haplotype 1 contain only 6 accessions, including our resistant parent and DCMP49. So which means not only this variety, there are other 5 varieties that we can potentially use for the resistant breeding program against XOC infection. And we also perform the protein domain prediction and protein-protein interaction, because as a typical wall-associated kinase gene, it should have the extracellular protein domain to sense the signal, and there should be a transmembrane region that can relay signal to the cellular regions, and intracellular protein domain for further relaying signals to the downstream pathway. So as our prediction, our WEX16 has the extracellular domains like GUB WEC binding domains and epidermal growth factor domains to receive the signals. And also for relaying the signal, we have we got the transmembrane region in our WEC16 and serine threonine protein kinase as a intracellular protein domain. And we also predict the possible protein-protein interaction. Here is our WEC16, and the proteins that highlighted here are the other proteins that might have interaction with the WEC16. So how, to which, what are these proteins? So these proteins mostly belong to MAPK cascade or MAPK signaling pathway. So we have, although we need further investigation, we got the idea that like the other typical WEC16, it has possible interaction with the MAPK cascade or signaling pathway. And we also performed the functional validation of WEC16 by having the CRISPR-Cas gene edited lines. We have got 2 edited lines here, with the A insertion or T insertion. And we also see the phenotypes of these gene edited lines, and we see that the disease symptoms show more severe or longer lesions when compared to the wild type. So which means knocking out of this WEC16 might give the plants to have weaker response to Ecto-C infection. And we also see the protein structure of these edited lines. As I mentioned before, for a typical wall-associated kinase, we should have all these kind of domains. But in our knockout lines, our knockout lines show that only transmembrane region and one of the extracellular domains, and they lost the intracellular protein kinase, which might be the reason that they cannot really signal to the downstream pathway and cannot activate the resistance mechanism. So from this experiment, our QTL-seq approach identify WEC16 as our potential candidate, and our expression data also show that this gene is very highly upregulated in our resistant parent. And our marker development validation also show that this gene might be useful for the breeding program, as well as our gene edited knockout line shows the possible function of WEC16. However, we need to have further investigations or further validations, like what are the real proteins that are interacting with WEC16 downstream, or what kind of genes are up or down regulated. Because when the pathogens try to invade the rice plants, the bacteria, they elicit some molecules like we call pathogen-associated molecular patterns. For example, like enzymes like patinase, which can degrade plant cell components pectin into these green things, which we call oligoglyceronides or OGs. This kind of degradation can be sensed by like other wall-associated kinase. These OGs can be sensed by WEC16, and they really signal through the transmembrane protein to the serine threonine protein kinase. And according to other WEC16 genes, we expect that this WEC16 might also interact with the downstream MAPK signaling cascade, or what are the expression of other defense-related genes, and what are the defense response, like a hypersensitive response and cell modification as stomatoconductance and the hours accumulation. All these information needs further investigation. And yeah, I would like to stop my talk here. And last but not the least is I would like to extend my sincere gratitude to my funding organizations, Thailand Scholarship 2022, the 2023 Crawford Fund International Engagement Award, and PhD research scholarship by SIACA, and my advisory committee, and the Right Science Center, Kasetsart University. And thank you for your kind attention.